Cancers such as leukemia are notoriously hard to treat, but a better understanding of what causes and drives their growth could open up new avenues for cancer therapy. That prospect has underpinned much of Dr. Stephin Vervoort's research over the past decade. Here at WeHi in Melbourne, he heads a lab that studies gene regulation, the systems that control when and how genes are expressed.
This process is critical for our normal development and how we respond to our environment. And when that process gets disrupted, that can cause human diseases.
Dr. Vervoort focuses on a key component of gene regulation known as RNA Polymerase II. Much like a photocopier, this enzyme reads our genetic code and sends faithful copies of it as messages to our cells in a process known as transcription. These messages act as instructions, directing processes within the cell.
When this photocopier malfunctions, that's when things start to go wrong. And really importantly, that fuels oncogenic or cancer growth.
Dr. Vervoort's team discovered that the transcription process has several distinct checkpoints.
RNA Polymerase II can do about 2,000 to 3,000 base pairs per minute, so it goes really, really quick. But it also needs to ensure that there is no mistakes in the copying of that message. Those checkpoints are there to ensure that at each point, everything is still okay.
When a checkpoint fails or is compromised, that can lead to errors in the messages sent. One example that Dr. Vervoort has studied is CDK12, a signalling molecule that regulates one of these checkpoints.
And when this one is lost in solid cancers, pretty much RNA polymerase loses the possibility to make long genes in our cells. And the long genes happen to be heavily involved in fixing DNA damage. And so when that DNA damage response doesn't work, you accumulate mutations, and that then results in cancer.
It's fundamental research that could help pave the way for novel anti-cancer drugs that block malfunctioning parts of the transcription process.
We've now made discoveries around this CDK12-related checkpoint, for example, where we've identified critical genes that are causing resistance and sensitization. And we believe that those might be prime candidates for therapeutic targeting down the line, and we're currently exploring opportunities to actually go down the route of drug discovery.
Originally from the Netherlands, Dr. Vervoort moved to Australia after completing his PhD.
When I finished my PhD, I felt like I can go anywhere in the world, but where do I really want to work? That's what drew me back into Australia, just the possibility to do groundbreaking science, but maybe even more importantly, do it in the Australian way, I would call it. Australians have a really good way of having fun, doing what they love, but doing it in an exceptional and thorough way anyway.
For his impactful research in human genetics, Dr. Vervoort has received awards including the Snow Medical Fellowship and the CSL Centenary Fellowship. He now adds the prestigious Ruth Stevens Gani Medal from the Australian Academy of Science to his growing list of accolades.